Hey, in this video we wanted to talk about one of the mechanisms by which we can observe horizontal gene transfer. And the mechanism we want to specifically address in this video is transformation. So let's begin by refreshing our memory about what it actually means to observe horizontal gene transfer. So horizontal gene transfer is a mechanism of gene transfer, and I want to contrast two forms of gene transfer. Let's start with vertical. So vertical gene transfer is a mechanism by which one cell can take its genetic material, copy it, and then pass it down to two daughter cells. So vertical gene transfer passes genetic material from parent to offspring. This is the kind of genetic transfer that happens in humans. This is the most common form of genetic transfer in bacterial populations, as it happens when bacteria reproduce. The thing about vertical gene transfer is that vertical gene transfer creates genetically identical offspring. So these two bacterial cells that are the result of vertical gene transfer from this parent cell have the exact same genetics as one another and as the parent. So while vertical gene transfer is particularly important, it doesn't really, it's not very exciting in that it doesn't create new kinds of organisms. All right, so again, vertical gene transfer, very important, but we want to talk about a form of gene transfer that's maybe a little more interesting because it creates more diversity in the world of living things. So we're going to talk about horizontal gene transfer. Horizontal gene transfer works differently. Here, a donor cell is going to give up a little bit of DNA, and that DNA is going to get added to a recipient cell. So this cell starts without this DNA and finishes the process with this DNA. So in the process of horizontal gene transfer, we change this recipient cell. It gains new traits. So horizontal gene transfer passes genetic material from a donor to a recipient. That will always be true. And the exciting thing about horizontal gene transfer is that horizontal gene transfer creates organisms with new genetic traits. Again, this cell now has DNA that it did not have before, and that may allow the cell to do something new. So again, we want to think about how horizontal gene transfer can occur. And it can occur in several ways, but in this video we want to address the mechanism of transformation. So let's check out transformation. So again, transformation is a form of horizontal gene transfer. Again, horizontal gene transfer requires two cells. First, it requires a donor cell. So I've made a very simple coccus-shaped bacterial cell here, and it's got its chromosome inside of it. We'll also need a recipient cell. So here I've made a very simple bacillus-shaped cell to demonstrate that the two cells are different from one another. And then this cell also has its own chromosome. So both of these cells have all the genetic material they need to carry out their everyday lives. Again, in transformation, DNA from this donor cell is going to end up inside this recipient cell. The question becomes, how? So the first thing that happens for transformation to occur is that this donor cell has to die. And when bacterial cells die, they undergo lysis. Basically, that means they fall apart, right? They pop. So the contents of the cell are released out into the world. So that's the next step. We've got the lysis. So I've just shown the membrane, the cell envelope of the bacteria has been destroyed. And now this naked DNA, the DNA that's inside the cell, is released into the environment. And we call it naked DNA because it's no longer surrounded by an intact membrane. In fact, let's just move it outside of the cell altogether. So again, the cell died, it lysed, and then the DNA that was inside it is released into the environment as naked DNA. When naked DNA is floating around in the environment, it will inevitably get broken into small pieces. So I'm just going to demonstrate that here, right? Over time, this DNA will break apart, but DNA is relatively stable. So the pieces of this DNA can persist in the environment for very long periods of time. So we had our donor cell die, it released its naked DNA. That DNA may or may not break into smaller pieces. And then in the next step, our recipient cell is going to pick up some of this naked DNA from the environment. So we'll show that here. One of these pieces of DNA can enter our recipient cell. And again, we just changed this recipient cell. It still has all its original genetic information, but it's gained some DNA that was originally part of this donor cell. This donor cell may have had different characteristics than the recipient cell. So this piece of DNA may tell the recipient cell how to do something that it could not do before. And that's the amazing power of horizontal gene transfer, is again, this recipient cell may be changed in terms of its phenotype, what it's capable of doing, because of this horizontal gene transfer event. So let me point out just a couple last details about transformation. Again, the process is really simple. A donor cell dies, its DNA is released into the environment as naked DNA, and then the recipient cell can pick up that naked DNA and then potentially utilize that DNA. So, one thing about the recipient cell, not all cells are capable of picking up naked DNA from the environment. And we have a special name for the cells that can pick up naked DNA from the environment. We call them competent cells. So only competent cells can pick up naked DNA from the environment. So you have to have a special characteristic to be able to do that. Basically, competent cells require some sort of protein transporter in the cell envelope so that the DNA, which is a really large molecule, can get across those layers, the cell membrane and the cell wall, and potentially the outer membrane as well. So again, to be able to pick up DNA from the environment, the cell has to be special in a certain way. And again, it has to be competent, meaning that it has transporters in its membrane that will allow it to move DNA from the outside of the cell to the inside of the cell. The other thing I want to point out is that with transformation, the donor cell and the recipient cell can be separated by significant time or distance. So remember, the first part of this transformation process is that the donor cell must die. But again, the DNA is particularly stable. So we have a dead donor cell, but the DNA is stable. So that DNA can last in the environment for a very long period of time. A recipient cell may not pick it up for months or days or years, right? The other thing is that with this stable DNA in the environment, it's possible that it could get moved around. If it gets put into a body of water in a river, it could start in one place and end up in a different place. So bacterial competent cells are potentially capable of, again, picking up DNA from an organism that lived a long time ago, or picking up DNA from an organism that lived far, far away from where it is. And that's unique or interesting about the transformation process. So we'll stop there. Again, that's one mechanism of horizontal gene transfer. We call it transformation. And if you have any questions about the process, I hope you'll ask. And otherwise, I'll talk to you again very soon.